All right, ladies and gentlemen, this is going to be a video lecture on a process called meiosis. Meiosis is the process that some cells go through to become gametes or sex cells. Um, I strongly suggest that you watch the meiosis animation video that I posted on Canvas. You have to watch that video, preferably more than once, and take good notes on it because it has great visuals and does a wonderful job of showing you how it happens. Um, so make sure you take good notes on that video. Make sure you take good notes here. And if you have any questions, of course, make sure you ask. All right, let's see here. So these are the goals for this section here. We want to make sure that we understand that normal body cells have a set number of chromosomes, and sex cells or gametes should have half that number. So for example, human body cells should have 46 chromosomes, but sex cells such as sperm and egg should only have 23. Meiosis is going to make that happen. The other goal is to make sure you know the different steps in meiosis. They are very similar to mitosis, but again, you need to make sure you understand those different steps. You should be able to compare and contrast meiosis to mitosis as well. And then finally, at the end, hopefully you can see how alleles from different genes can still be inherited together depending on where they're located on the chromosome. And this idea is a lot easier for me to show you than it is for me to describe without pictures. So I'll just leave that until we get to the pictures for that one. All right, so there's a little bit of vocab we got to get down first. So again, the purpose of meiosis is to take a cell with a certain set of chromosomes and divide it so that in the end it ends up with half the number of those chromosomes. So before we start, we need to go over, like I said, some vocab. The first term is homologous. According to your textbook, homologous are chromosomes with the same genes, one originally from each of the organism's parents. In other words, these are chromosomes that are partners. And if you recall, I told you that our chromosomes and our genes work in pairs. The reason being is that you get one set of genes from mom and one set of genes from dad. That's the same exact thing as saying if you get one chromosome from mom, you have to get a partner chromosome from dad. So if you take a look at the diagram here, I know by looking that this large red chromosome is homologous to this yellow chromosome, here, the big one. That means that these two chromosomes are partners and they have the same types of genes on them. One came from one parent and the other one came from another parent. Same thing with these two smaller ones here. I know by looking that these two are homologous because they're the same size and the same shape. And so what that means is that if they're homologous to each other, the genes on this little chromosome should be the same types of genes on this little chromosome over here. One came from one parent and the other one came from the other parent. So that's what homologous is. The next vocab term is diploid. Di means two. Diploid, according to your textbook, is containing both sets of homologous chromosomes, also symbolized by 2N. A diploid cell would be a normal body cell that has two sets of chromosomes. If you remember, one from mom, one from dad, right? So your normal body cells are diploid. They have the two sets of chromosomes. Haploid is the next term, containing only a single set of chromosomes, which is symbolized by 1N. In other words, haploid is a haploid cell is a cell that has only one set of chromosomes, or you could say a cell that has half the number of chromosomes than the other normal body cells. So the only haploid cells an organism should have are sex cells, gametes. Those are the only cells that you should have that are haploid. All right. Before we start, I also forgot to point out, you need to make sure that you pay attention to the number of chromosomes we're starting out with. So for this cell, and as we go through meiosis, you need to note that we are starting with 4 chromosomes here. 1, 2, 3, and 4. All right. Also, these chromosomes are duplicated. So this is actually one single chromosome here, and this is its exact replica right there. All right, so the chromosomes have been replicated already. All right, so before we begin going over meiosis, let me just quickly review mitosis first. If you recall, the point of mitosis was for your normal body cells to divide into two, because maybe they needed you need more cells. Maybe some of your old cells are breaking down or dying. So the process of mitosis is simply to produce more cells that are identical to each other. So if you look, here's a cell. There's 4 chromosomes here, and this cell is going to go through the different phases of mitosis here. If you recall, the first step in mitosis is prophase. All right, so here's prophase. Metaphase, M for metaphase, M for middle. So these replicated chromosomes get moved to the middle by the spindle apparatus, right? And then the sister chromatids are split apart or split away from each other. And we said, all right, well, we could remember it by saying A for anaphase, A for away or apart. So here they are, the sister chromatids being split apart. And then finally in telophase, we said, all right, let's say T for telophase and T for 2 new nuclei. And so here are your 2 brand new cells. They should have 4 chromosomes each, and then now you have 2 new identical cells. So that's mitosis. Meiosis is going to be different. There's a lot of similarities, don't get me wrong, but the end result hopefully will be different than mitosis. So let me give you a real quick rundown of meiosis. We're going to start right here. You start out with a cell. In this case, again, we're starting out with a cell that has 4 chromosomes. Take a look. 1, 2, 3, 4. So you got 4 chromosomes. If you'll notice, the homologous chromosomes, they pair up. In other words, they find their partner, they pair up, and this weird thing called crossing over occurs. I will go over what this is and why it is so important in the end. Sorry. All right. And then once these homologous pairs pair up with each other, they are moved to the middle during metaphase 1. Anaphase 1, they're gonna get separated, so the homologous pairs are getting separated from each other. And then finally, during telophase 1 in cytokinesis, you see 2 new cells that have formed. And I want you to notice, because this is important, these 2 new cells, if you look, there's only 2 chromosomes in each of these cells now. So if you look, this one only has 2 chromosomes, and this one over here only has 2 chromosomes, right? We started with 4 chromosomes. We are now ending with 2 chromosomes. All right, so that is the end of meiosis 1, but we're not done yet. There's also a meiosis 2. So once this cell splits into 2, and we cut down the number of chromosomes to half of what it was, we go on to meiosis 2. See, it says meiosis 2 there. All right, so here are our 2 new cells that just formed. Please notice that there's 2 chromosomes in each one. All right, and we go into prophase 2. It's going to be very similar to prophase period, but here's prophase 2. The spindles are forming. There's the 2 chromosomes, right? Then we go on to metaphase 2. M for metaphase, M for middle. the chromosomes will be pulled or moved to the middle by the spindle apparatus. So here they are lined up in the middle, both cells. Then we go into anaphase 2, A for anaphase, A for apart. Now, pay attention. The sister chromatids are now being split apart, right? So now these are single chromosomes now. All right, if you take a look, single chromosomes, same thing here. So the sister chromatids are being pulled apart now by the spindle in both cells. And then finally we go into telophase 2 and cytokinesis. This is where each of these cells is going to split into two again. And so we should end up with 4 new sex cells or gametes. And again, please note that each one of these new sex cells or gametes only has 2 single chromosomes in each one. Remember, we had originally started with 4 chromosomes. We now have only 2 chromosomes in each one, and that is the whole goal. That's the whole goal here, is just to end up with gametes that have half the number of chromosomes that we originally started with. Okay, so now just to review, we can go over each phase of meiosis in some detail here. So again, the whole point of meiosis is to take a cell and divide it so that the cells that you end up with have half the number of chromosomes that it originally started out with, because sex cells or gametes should only have half the number of chromosomes that the other normal body cells have. So if you are talking about meiosis in humans, what would happen is you would start out with a cell that has 46 chromosomes, and by the time that cell was completely done going through meiosis, you should end up with 4 new sex cells or gametes, and they should not have 46 chromosomes in them. They should have only 23 chromosomes. That would be half. So let's go over these phases in a little bit more detail. So we start with prophase 1. In prophase 1, the nucleus is obviously gone. It has broken down, and the spindle apparatus has started to form. These chromosomes are replicated. Here's for example one sister chromatid, or you could call this one chromosome if you want, and here is its copy. Alrighty. Also, very important in prophase 1, the homologous chromosomes pair up. In other words, the partner chromosomes pair up. They find their partner and they line up together. So these 2 chromosomes are homologous to each other. That means they're partners. They have the same types of genes on them. All right, same thing with these 2 little ones. These 2 are homologous to each other. That means that they are partners and they have the same types of genes on them as well. Friendly reminder, you could say that one of these came from maybe mom and the partner one came from dad. All right, now I really want to point out this very special step here, where if you take a look here, or we could look down here, but if you look, these 2 homologs, what's going to happen is when they pair up, they are going to literally cross over each other and swap pieces, and that is absolutely fine. You want that to happen. The reason this is important is because by the time you are completely done with meiosis, you will have 4 new gametes that are not genetically identical to each other. There's a little bit of variety, and that's exactly what we want. We want our gametes to be a little bit different from each other. We don't want our gametes to be all identical. So that is the purpose of crossing over. You still have to read up on this in your textbook so that you're able to identify and understand the textbook language. All right, so that's crossing over. It occurs during prophase 1. It's important. All right. If you remember from when we did mitosis, the next step after prophase is metaphase. Oh, Sorry, I forgot to point out the little centrioles that the spindles come out of there. So there's the centrioles, there are your spindles, and then of course, hopefully you remember that these are chromosomes. If not, it's okay, just make sure you review. And then the little center here, where they're attached, is called the centromere. All right. The next phase is metaphase. M for metaphase. M for middle. In metaphase 1, the homologous chromosomes will be moved or pulled to the middle by the spindle apparatus, and it's pretty simple and straightforward. Here are the homologs, there and there, and they have been pulled towards the middle by the spindle apparatus. And that's about it for metaphase 1. After metaphase is anaphase, A for anaphase, A for apart. And so during anaphase 1, the homologs, or you could say the homologous chromosomes, it means the same thing, but the homologs or the homologous chromosomes will be pulled apart from each other by the spindle apparatus. So here goes one chromosome in one direction, and then here goes its homologous partner in the other. Same thing with this pair here. Here is one chromosome being pulled to one end, and here is its partner or homolog being pulled to the other. And that's about it for anaphase 1. Again, please remember these are sister chromatids. Because of crossing over, this chromatid here is now no longer exactly identical to this chromatid here, and that's a great thing. We want that to happen so that we don't end up with identical gametes. After anaphase 1 is telophase 1. If you recall, we said T for telophase, T for 2, because 2 new nuclei are beginning to form, and that's what you see here. As a matter of fact, this cell is starting to split into 2, so you can see 2 new cells forming as well. So here are 2 new cells that are starting to form. Here are the 2 nuclei, and this is really important here, folks. If you'll notice, this nucleus only has 2 chromosomes in it, and this nucleus only has 2 chromosomes in it. If you recall, we originally started with 4. Now these 2 new cells only have 2, and that's exactly what we want to happen. We are now starting to cut that number of chromosomes in half. So that is telophase 1. These 2 cells will completely finish splitting apart. Excuse me, and that will then take us into prophase 2. Actually, it'll take us into the second division called meiosis 2. That's what we're going into. We're going into meiosis 2. Meiosis 2 begins with these 2 new cells that we just got, and the first step is prophase 2. So we are now in prophase 2. It's going to be very similar to the other prophase, but here again I want to point out, this is really important, these 2 new cells that we just got have only 2 chromosomes in here. So there's your prophase 2. After prophase 2 is metaphase 2, M for metaphase, M for middle. These 2 chromosomes in each cell are moved to the middle during metaphase 2 by the spindle apparatus here. After metaphase 2, we go into anaphase 2, A for anaphase, A for apart. So now the sister chromatids will be pulled apart or pulled away from each other by the spindle apparatus. So if you look, we have these 2 cells, they're going through anaphase 2, the sister chromatids are being pulled apart. So that's it. You go into the next phase, which is telophase 2 and cytokinesis. All right. So if you'll notice, by the time you get to telophase 2 and cytokinesis, you should have 4 new gametes or sex cells. Remember, we started out with 1 cell. It divided twice, so you end up with 4 new cells. And most importantly, Each one of these gametes or sex cells only has 2 single chromosomes in the nuclei. All right, and also please notice that we used color to represent this, but the 2 chromosomes in each of these cells are not identical to each other, right? So this chromosome is all red, this one is mostly red with a little bit yellow, and then, if you take a look, they're all a little bit different from each other. Again, you don't want your gametes to all be identical. All right, so the end product of meiosis is 4 daughter cells that have half the normal chromosome number that you started out with. If you have any questions, please be sure to write them down so you don't forget them, and then make sure that you get those questions answered before we take that test later. So that is the end of telophase 2 in cytokinesis, and this is also the end of meiosis. So real quick, here's a quick summary. We start here. Here's your cell that you're starting with. By the way, the cells that go through meiosis are called germ cells or germline cells. Those are special cells that are destined to become gametes or sex cells. So you start out with this germ cell here. In meiosis 1, you start out with prophase 1. Here are the homologous chromosomes that pair up. The crossing over occurs. All right, then you go through metaphase 1. The homologs line up in the middle. Then the homologous pairs are pulled apart by the spindle. And then finally the cell splits into 2. And this is where you go from 4 chromosomes to 2 chromosomes in each cell. So now you have 2 new cells, and here they are right up here. These are the 2 new cells with the 2 chromosomes. Now we begin meiosis 2, or the second division. It's going to be very similar to the first division. Here's your prophase 2. Here are your 2 chromosomes. Here's your spindle apparatus, right? After prophase 2 is metaphase 2, M for metaphase, M for middle. So the sister chromatids, you call them chromosomes too. It's going to mean the same thing, but the sister chromatids or the chromosomes are moved to the middle by the spindle. Then you go into anaphase 2, A for anaphase, A for apart. Now the sister chromatids will be pulled apart, and now you are going to have single chromosomes. Excuse me. And then finally, you go into telophase 2 in cytokinesis. So this cell will split into 2. This cell here will split into 2. And you will end up with 1, 2, 3, 4 new gametes. Again, note that these 4 new gametes each have only 2 single chromosomes in each one. And they are all a little bit different from each other. 4 haploid daughter cells. These will be the new gametes. And that is it for meiosis. Here's just a little side-by-side comparison of mitosis and meiosis here. You're more than welcome to peruse over that, but I think you get the point. Again, if you have any questions, you should probably write them down so you don't forget. But that's about it. And I think, take a look here. In mitosis, if you start out with 4 chromosomes, you better end up with 4 chromosomes. The number of chromosomes should not change. As a matter of fact, nothing should change. You should end up with 2 identical cells. However, in meiosis, if you start out with, say, 4 chromosomes, you should end up with half that number, which in this case would only be 2. So gametes or sex cells should only have 1 set or half the number of chromosomes.